don't worry again about getting too worried about which DNA polymerase does what. Just know that you'll have DNA polymerase that will extend the RNA primer, and then you'll have another DNA polymerase that will come in and replace the RNA primer with DNA nucleotides, which will have a different sugar, and then instead of uracil, you'll have thymine, and then your ligase will come in and connect all of those little Okazaki fragments. So that's how we get away with going in the wrong direction. We don't. We just go backwards and make the fragments in the right direction. Okay, this is a nice little graph. I told you that one of the hardest parts, other than understanding how we go in the 3' to 5' direction, the other challenge is to keep straight all the different enzymes. This one does go and it kind of tells you the difference between polymerase 3 and polymerase 1. I'm not going to hold you accountable for that. I'll just say DNA polymerase and just know that there's one DNA polymerase that extends the sequence and one that basically goes in and takes the RNA primers out and replaces it with DNA. But that's in your notes, just a nice little graph to study from. This is just a little note about how does this happen, because it's called these are all proteins that are interconnected with each other. Remember how we talked about proteins have these domains when we talked about our bioinformatics lab? Some of these domains help connect these proteins together, and they actually make what's called a DNA replication machine. They all work together in concert. And so at first they didn't know, is it that the replication machine is stationary and it's pulling the DNA through, or is the replication machinery moving along a strand of DNA? They weren't really sure. Most recent evidence actually points to the idea that the replication machine stays stationary and kind of stays bound to inside the nuclear lamina, that framework inside the nucleus. So they think that it kind of stays in place and then just reels the DNA through and replicates the DNA as it reels it through. Just a little side note about how we think this occurs. This is just pointing that out, all of these different enzymes that are involved, lagging strand, leading strand, this whole replication machine process. The machine is staying stationary and just pulling the DNA through. It's not a confirmed theory, though. It's just what the evidence currently supports. So that's all we're going to talk about with how we replicate it. And now, how do we make sure we replicated it correctly? Because one thing that they found is that when they looked, as the DNA is being replicated, they found that there's a much higher error rate than they see in completed DNA products. So what's going on in between replicating the genome and the final product? Why are we seeing a lot more errors in the replication process than we see in the two daughter strands that come off of the replication process? And so one of the things that occurs, first off, the DNA polymerase, as it's adding new nucleotides, has a proofreading function. So it's looking at this base pair and it's making sure that it's adding the correct complementary base pair. And if it adds the wrong base pair, it will basically take it out and put the correct one in. Just like if you're typing and you type in the wrong letter, you hit delete, go back a key, and then you put the correct letter in. So that's the first level of correction that occurs. So that's called mismatch repair of DNA. But you can also damage DNA by other materials. So exposure to certain chemicals, carcinogens, X-rays, UV radiation, all of these things can damage the DNA in your cell. And that, when I say that, I'm talking about all of your somatic cells. So right now we're not talking about gametes. We're just talking about, okay, a cell divided, and then you went out and you sat out in the sun for a long time, and some UV radiation created some damages in the cell and the DNA of your cell. If that is not corrected, every time that cell divides, the daughter cell will have the incorrect sequence, okay? Because, normally we think we can only pass down damaged DNA through our gametes. But if you do get an error or a mutation in a somatic cell, you're not going to have to worry, you're not going to pass that, you're not going to pass that defect or that incorrect sequence down to your children. But every time that cell divides, if it's not corrected, there's going to be that mutation that persists in that cell population. So there's additional machinery that's in place to say, like, oh, this person sat out in the sun too much, or this person ate a bunch of stuff that was cooked on a charcoal grill, or this person's been smoking cigarettes, or whatever it is that's causing the damage. There's a whole set of enzymes that are responsible for going in, taking out the damaged sequence and replacing it with the correct sequence. And there's actually a lot of cancers that you can inherit are mutations in the enzymes that correct DNA damage. So I don't know if you've ever heard about people who can't ever go out in the sun. There's a condition, trying to think, it's called, I've totally forgotten the name of it. Something pigmentosin, something pigmentosin. The name of it's not important, but basically it's a defect in one of these enzymes that if you get exposed to sunlight, there's a specific type of DNA damage that occurs called thymine dimers, and it's the enzyme that's responsible for fixing that doesn't exist. They have a genetic mutation that they don't have the correct enzyme. That, of course, if it's a genetic mutation that you can pass down, it's in your gametes, that's a problem for your children. Because if they inherit that mutation in that enzyme, then if they go out in the sun and they get a mutation, that mutation is never going to get corrected, which means you have a much greater risk of having skin cancer with much less exposure to the sun at a much earlier age than you would normally expect to see it. So that process, when we go in and we find that there's damage that's been caused by some type of chemical or physical agent and we want to fix it, it's called nucleotide excision repair. So we have another enzyme called nuclease. It's going to come in and remove the damaged nucleotides, and then DNA polymerase can go in and put the correct nucleotides back in sequence. And so here's just a little image looking at that. I told you one of the things that happens with sun exposure, one of the damages to DNA that occurs when you expose yourself to the ultraviolet radiation from the sun are called thymine dimers. And a thymine dimer is going to occur if you have DNA that has two T's in a row. So T. The T should be bound to what? They should be bound to
A.
But in this case, what happens with UV light, this bond breaks and they form a bond with each other, and that causes the DNA to buckle, and it makes it, creates some damaged DNA that's hard to replicate. And again, this is one of the things that can lead to accumulation of mutations in certain genes that can cause skin cancer. So we want to fix that. So we've got this little thymine dimer that's caused the DNA to buckle. The thymine is bound to itself instead of being bound to its corresponding base pair. So this nuclease enzyme is going to come in, it's going to cut out the damaged DNA, and then DNA polymerase now has a nice 3' end here that it can attach new DNA nucleotides to, but it can't bind those last two together, so you're going to still need that little ligase enzyme to connect the newly synthesized DNA from the DNA polymerase. So this is why you can have damage to your genetic material in your cells, in your somatic cells, and this is why usually people don't get cancer that's not genetically based until they're a lot older. Your risk of cancer goes up as you age, and it's because we have a lot of machinery that can correct damages as you go, but over time, as you continually expose yourself to things that are harmful to DNA, things with high energy, radiation. Tennessee, we've got a lot of radon. If you're up in the high atmosphere, you're exposed to more high energy radio, what's the term I'm thinking for? Not radiation. High energy things that can damage DNA. I can't think of the word right now. But there's a lot of things that we do to our bodies, a lot of things that we eat. There's a lot of toxins, pollutants, chemicals that can cause damage to DNA. So the older you get, the more likely you are that there's some mutation that happens in your genome that could be problematic. It should be a mutation in one of these enzymes that corrects DNA. It could be a mutation in one of the enzymes that we talked about. It's one of the clock enzymes that's important to allow mitosis to occur. There's certain specific genes that if you get mutations in them, they're highly associated with becoming cancerous. So we want to repair mutations as they occur. But at a certain point, you can overwhelm the system. If you get like a really high dose of radiation, you were around Chernobyl, or you were around from Fukushima when the nuclear reactor blew up. You can get these high levels of radiation exposure that your machinery, your repair machinery can't keep up with the number, the amount of damage that is done.